Welcome to Everything STEAM. I'm your host, Sam Stanford. As a physicist and structural engineer with Jacobs Engineering, I've made many connections with some bright individuals who are either working, studying, or self-taught and passionate about our particular topics of discussion. In this episode, we plan to talk about a field in science that has exponentially progressed since it was founded in the mid to late 1800s by Gregor Mendel. This field is, of course, genetics. Once we get through some of the basics like DNA, genome sequencing, gene expression, and epigenetics, we'll highlight the exponential growth of this field by discussing a very exciting, practical, and ethically challenging method of genetic editing called CRISPR Cas9 that has profound implications of curing our fellow humans from diseases such as sickle cell anemia. If you've listened to one of my podcasts before, you know that this is not in my wheelhouse, so I brought in a lovely guest that is well equipped in this realm. So without further delay, let me introduce you to Brenna Stanford. Brenna is a molecular ecologist working on her PhD at the University of Calgary, Alberta. She grew up on a small island on the west coast where she developed a deep curiosity and love for the beautiful complexity of the natural world. In 2015, Brenna graduated with an honors degree in biochemistry from the University of Victoria and then worked as a conservation biologist for Parks Canada before starting graduate school. Over the last 7 years, Brenna has done research on a number of different species, broadly focusing on using molecular techniques to better understand how organisms respond to and adapt to their ever-changing environment. Brenna then completed an MSc in Evolutionary Biology in 2019, where she used whole genome sequencing to answer conservation questions for the endangered Banff Springs snail. Currently, she's near completion of her PhD, where Brenna has combined her passion for molecular processes and conservation by investigating the ecology of gene expression to test predictions about the resiliency of species to climate change. So, now that you've been introduced to Brenna and the topic of this podcast, we're going to head into the first segment where we plan to relay some basics in the field of genetics. Brenna, welcome to the podcast. How are you?
Hello, I'm great. How are you doing?
I'm good. It's kind of funny. Whenever I reached out to you on TikTok and we kind of had a back and forth, didn't realize we didn't realize that both of our last names were Stanford until we met.
The joys of connecting with people on social media where you're not using your actual name.
Yeah, I would really love to know the odds of meeting someone in this kind of realm of science communication, just and having the same last name. I thought it was funny.
It was funny. I went to a conference last summer, and there's a prominent lab in what I research at Stanford. And the person looked at my name and was talking to me thinking I was one of his grad students. And I was like, I don't know when to interject that I don't go to Stanford. That's just my name. So it was very funny. I was like, oh no, I go to University of Calgary. And he's like, where? I was like, yeah, that's fair.
Oh man, that's that institution biases runs way too deep.
Yeah. I mean, it's really more important about who your supervisor is, but
yeah, I agree with that. So, okay, we're talking about genetics, obviously, and
one of my favorite topics.
That's right. But before we get into anything too, I guess surface level, then deeper later into the podcast, I want to start out by just asking you, why did you really get into biology? It was just because you love Punnett squares, or what?
Oh yeah, I love me a good Punnett square. Just no, oh man, I was talking to my best friend actually a little while ago, and she was like, and she's known me since I was 6 months, and she's like, I just don't know what you would have been if you weren't a scientist. She's like, it just nothing else really makes sense for you. But I, as a common thing of students in biology, I thought I wanted to be a medical doctor. And so I went to college, which if there's anything you can get from this, college is a wonderful option for the first 2 years of your degree, at least in Canada. I don't know so much about in the United States. But I went and I had a wonderful program, applied chemistry and biotechnology. But then I took a second-year biochemistry course with a phenomenal prof, and I fell in love. And so when I transferred to university, I ended up doing a degree in biochemistry. My parents have always been so supportive of whatever I want to do, but I think that this was the first thing that my mom was like, well, maybe you should just take the MCAT. And I was like, no, I really don't think I want to be a medical doctor anymore. Finished an undergrad, did an honors project where I studied the function of a gene, which we'll get into more about. But then realized I didn't want to study one protein for the rest of my life. So I took a bit of time off, was a ski bum for a little bit, and started a master's with Parks Canada doing a whole genome conservation sequencing project in an endangered species. Through that project, met my current supervisor, and now I'm doing a PhD broadly looking at how species adapt and persist in their environment, and specifically as environment changes. And I study that using a little species called the three-spine stickleback. And I don't know how nerdy this is, but got a little tattoo of the guy. It's a little species of fish. And
I love that.
Yeah. So that's currently where I'm at. I'm finishing up my PhD in this project and looking for postdocs. Yeah. But I love DNA. And I don't know. I just was like, yeah, I was learning about how like biochemical pathways worked and how DNA expression worked and all these things. And I was like, oh my God, this is amazing. And everyone else around me is going, oh God, I can't wait for this course to be over. And I'm like, oh. So it's amazing when you like go to university or like find the thing that like kind of clicks for you. And then like really being able to apply that in my current position to like ecological and evolutionary questions has just been a really wonderful fit.
Oh, 100%. But today, so for this segment, we're going to kind of set some, maybe like start a foundation for our later topics. And we want to, I guess, address, I'll just direct it towards you because you're the expert. I'm not even going to attempt this, but we talked offset about starting this out by running through like cell to DNA in stages. So
yeah,
take it away.
Okay. So when we start talking about DNA, RNA, and all of these terms, it's important to kind of think about how this is packaged in your body. So your body has made out, hopefully this is not a shock to anybody, but made up of a lot of different cells. And so what you have is cells that have been differentiated into different kinds. So you have skin cells, you've got like, for example, your muscle is made up of, I believe, like 13 different cell types. Wow. And so these are all stem cells that have been cued and have differentiated into these different types of cells that you need to make you. Right? Because you can imagine that the job of an eyeball cell or a specific part of your eyeball is a very different job than your skin cell. So it needs different characteristics. And so stem cells are unprogrammed cells that sit there and then they get a cue from the environment and then they become a certain cell type. So that's important when we start talking about DNA. Because the information to make all those different cell types and to create all those functions exists in every single one of your cells. But there are other factors that allow it then to become certain different types and have these different functions. And so when you kind of go into a actual just cell, I'm doing the like circle with my hand. Like I'm sure that people have seen this. It's like it's a circle like this, and then there's a little circle inside, and then there's some mitochondrial drawn, and it's all very clean and unchaotic. That's not, of course, not what real biology looks like. I was frankly quite upset when I learned this. I was like, "What do you mean? It's not this beautiful, clean little drawing we have." But the kind of two main structures that we'll talk about, probably I think that's important for this conversation, is that there's two kind of broad zones in a cell. There's your circle, and all of that in there is called the cytoplasm. And then on the inside, there's the smaller circle, which is called the nucleus. The nucleus has your DNA. and it's kind of like a vault. It keeps your DNA, and not a lot of information is ever allowed to go into that area because, as you can imagine, your DNA is very valuable. It's got the instructions of how to make everything that makes you function. So
Yeah.
we've got to protect that. And then the cytoplasm is where like all of the machinery, the mitochondria, ribosomes, and a bunch of other things exist. But there's these two kind of like, there's the membrane phospholipid bilayer that sits at the outside of your cell. So there's the outside of the cell, inside the cell, cytoplasm, and then your nucleus, which has your DNA in it. So that's kind of the structure of a cell for eukaryotes, which is what we'll talk about today.
Right. Yeah.
As cool as bacteria are, we're not gonna get into those today.
Right. And I kind of want to just
unless
say,
you want to ask.
That's okay. We can keep it simple. I do want to kind of highlight this quick little fun fact. I think there's between 30 and 40 trillion cells at one time in the human body, or that make up the human body.
About right.
So that's right. If you can think about, if you can think in terms of millions, which most people cannot, that is 30 million, and then take that times 1,000 and times 1,000, and that's what you get.
Is that nuts?
That's pretty crazy.
And that's just existing to make you.
Right. And I think there's also that really cutesy like fun fact as well, is like, isn't it every 7 to 10 years your body completely cycles through all of its cells or something of that matter? Is that true or not?
I feel like you're really putting me on the spot here. I haven't looked that up. I feel like I just heard that when I was like a teen and was like, yeah, sounds about right. And
yeah, I don't
know. I feel like now I gotta look it up.
Okay. Yeah. So do your research. Fact check me. Let me know.
That sounds about right. Because like your body is constantly, I think that actually is a really important point, is that your cells have a limited lifetime. And so your cells are copying themselves to make another cell to take its place. Because then there's something called, like, there's programmed cell death, where the cell is like, okay, I've lived a good run. I've got some cues. It's a good, like, we're going to dismantle all of this and we're going to do it in a really like clean and orderly way. We're going to kind of dismantle ourselves. We've made a new one. We're good. Then there's like unprogrammed cells death, which is kind of like a cell bursting, where all of these kind of, it's a much more chaotic process, and it's generally based off of a reaction to something. And so when it's copying itself, you have to copy your DNA over. You've got to make an entire new copy of it. And so your cell has really sophisticated machinery to like copy all of the DNA and do this in a really, like, as you could imagine, it could be easy to make mistakes. And I think that people don't quite understand, maybe that's unfair. I know that I was surprised to figure out just how big DNA is. It's huge. Like, if we're just talking about humans, in every single one of those trillion cells you have 6.5 feet, about 2 meters of DNA. Exactly. I know. My Canadian isn't showing here. I am trying to talk to your American audience. So 2 meters of DNA so it's this really, really thin string that's packaged into the nucleus of every single one of your cells. And like, to kind of put that into perspective, if we think of DNA, we can get, or I realize I'm kind of jumping the gun here a little bit, but if we consider it as like words, it would take someone typing, I want to make sure I get this correct, 60 words per minute, 8 hours a day. It would take them 50 years to type out all the information that you have in one of your cells.
Oh, wow. I think
Isn't
I can
that
add
crazy?
another. Yeah, no, it is kind of, it is very crazy. Based on like the, we want to just stay on a base level for right now. If you were to cite the letters in the human genome, if you were to say one letter per second, you would be doing that for 209 years.
Oh my goodness, that's a great way to think about it.
Yeah, it was 6.6 billion letters. Yeah. Or yeah.
Well, if you consider dual copy, 3.2 billion. Yeah.
Right. Right, exactly. That's still crazy. Okay, I guess cut it in half. You're still at 100, a hundred
Oh,
and four and a half
very
years.
feasible now.
Yeah, no worries, no worries. Just dedicate your whole life, and
And hope that you live over 100.
right, right, which not most of us can do.
Well, not yet. We'll get into more about CRISPR. So,
Ooh, exciting. So you had a really interesting analogy, I think, that you were getting to whenever you were talking about the nucleus being the vault of the cell. So would you
mhm.
mind jumping into that a
Yeah.
little bit more?
And I realized I kind of jumped ahead about talking about letters of DNA.
Mhm.
So DNA is made up of 4 different molecules that we would consider. So it's A, T, C, and V. And this is the like name for the molecules of it. And so DNA is structured in different combinations of these letters. So if you kind of zoomed into that 6.5 feet or 2 meters of DNA, I don't know why I keep using feet. You zoomed in to a small region and looked at the letters of
Mhm.
it. It would be something, maybe it would be like AA, T, G, CCC, and it would be different combinations of these.
Mhm.
And those different combinations code information. And it's different information depending on the order in which these letters are. And so
Right.
that is going to get important when we start talking about genes. And so the A, T, C's, and G's, I like to think of it kind of like it's a book. It's an instruction manual, a huge, huge instruction manual that is all of the instructions on how to build and run a human. And obviously, this can get expanded to an organism of any kind, but I feel like
Right.
we're talking mostly about humans. So it's how to build and how to continue functioning this human. And
Mhm.
so if we think about like a human or a species or an organism as like a house, it has how to lay the foundation, when to build the walls, the fact that you need to build the walls before you do any of the inside framing,
Mhm.
put the roof on. My dad's a carpenter. So,
So is mine.
um, oh, fantastic. And so it has all of that information in it.
Mhm.
And that sits in the vault. But as you can kind of think, you don't need all of that information all of the time. So, and if we think about it, maybe if we think your eyeball is a window, So you need glass panes, you need how to like make a little frame, you need to know all of these things. But you don't need to know how to build a kitchen table to build a window. So, but all that information of how to do everything is in every single one of your cells. So that is sitting in the vault, all of that
Mhm.
information. What happens is that there will be in your cell a little cue that says, hey, I think we need to build a window. And so through a series of like kind of telephone, which is like signal transduction, so goes through the outside membrane of the cell and goes through the cytoplasm through a bunch of communication. It hits the front desk of the nucleus, the vault, and it
Mhm.
says, hey, I got a request. I need to make a window. And the secretary is like, I got you. Or I guess the librarian. I don't know. We're mixing analogies at this point. And it goes to the giant instruction manual and it flips through and it finds the instructions on how to build the frame of a window, how to source glass. And it copies those. It makes a bunch of different copies of it, like photocopies that region. And then it brings back, because are you going to give someone the entire thing, all 2 meters of it, or like this huge book, or just the specific instructions? So you give the specific instructions, that gets slid back over the desk, and that now goes into the cytoplasm,
Mhm.
where there's machinery on how to build everything that you need. And so if we consider the vault that contains DNA in that space, so like the instruction manual is the DNA, those little photocopies is what we would consider RNA. So it's the instructions that have been copied from the DNA that now go into the cytoplasm and that are specific for certain functions and how to build very specific things rather than all of the instructions on how to build a human and run it. I don't know if that made any sense. I'm hoping.
I think that's a really great analogy, honestly, like for a layperson. It's
perfect.
probably the best that I've ever heard. Honestly. Do we also want to cover what RNA is with respect to DNA, like in terms of how they're structured? Because I think that's important.
So DNA is packaged in what we call a double helix. If we consider like one side of the double helix, it's an A, a T, a G, let's say. A binds T on the other side, so it will be a complementary strand. So they bind across, and then they twist, and they create this double helix shape. And that's packaged into chromosomes, into your nucleus. Because remember, you got to package 2 meters of this stuff into a very small region. So that's also like part of what the DNA, when the little memo comes and says, hey, we need this instruction, it goes through, files through all of that 2 meters and finds the region that has that instructions and actually opens it up, cuts it, like basically breaks apart the A and T from each other, makes it open, and then it copies that into RNA. And so the RNA is actually only single-stranded. It doesn't have a complementary pair. It also has a different base. So it has a U in it.
Instead of the T, right?
Instead of the T.
U, A, C, and G, right?
Yeah. And so that gets, and then that kind of comes off of the DNA. This gets sealed back up because it's very stable. It's very stable when it's bound to itself. And we like that because we don't want to damage our DNA in any way. But then the RNA floats back out of the nucleus into the cytoplasm, and it gets read and built into whatever it needs to be built. And then because it's a little bit unstable, because it's in this single-stranded state, and also because there's some things, it gets degraded, which is good because you don't need the instructions. Once you've built the window, you don't need the instructions to build the window sitting around, taking up space. And so it gets degraded, and then the parts of it gets recycled into making a new RNA. Yeah.
Makes sense. That's pretty cool. One cute analogy, I think, or not even analogy, one cute saying for the for what the base pairs, right? In the helix, the double helix, is apple in the tree, so A and T, and then car in the garage, C and G. I think.
Oh, that's precious.
Yeah. Oh, yeah. Okay.
I never heard that.
Oh, there you go.
Gosh, I'm going to use that with my students.
Yes, there we go. There we go. I learned that forever ago, and it has just kind of stuck in my brain for some reason. Whenever I was getting ready for this podcast, it came back to me, and I'm just like, like I have to use this.
Well, thank you for sharing. I very much appreciate it because I don't know, it's like always nice to have things that make it a little bit clearer for people to remember, because it is complicated, and it's something that I've been learning about for 13 years now, and I'm still like, okay, sometimes, so it's great.
No, it's really cool. Okay, so I think moving on, we have we talked about the base pairs, the nucleotides. Is that right? Is that the right term for them? So so the nucleotides, which apple in the tree, car in the garage. We go.
I do love that.
Yes. So we go to DNA, and then we take a step up from that, and we're at genes. So what is a gene?
Okay, well, that is a loaded question, actually, because in one of my undergrad courses, we had a conversation, what is a gene? And it was sparked a debate, which I think is interesting when we think about language use in science, because a lot of the time we use the same terms and we have different meanings, or not even different meanings, maybe that's unfair to say. we have slightly different opinions or ideas that brings it up. So my background is biochem, as I've mentioned, and I work with a bunch of ecologists, and we have different ideas about molecular. Anyways, that didn't answer your question at all. So a gene, traditionally, and you'll hear me sometimes maybe say a protein coding gene. So traditionally, a protein coding gene is what was considered a gene. So you may have heard the thing of like, oh, 98% of your genome is junk if you're of a certain generation. And this was the kind of theory until I think probably 15 years ago, is that it was called junk DNA, anything that didn't code for a protein. So when I say code for a protein, going back to our building a house analogy, and where we were just talking about copying certain regions. So what I was talking about with that is what we would consider a protein coding gene. So the memo, or like the photocopied instructions, go out into the cytoplasm, and then machinery, which is called a ribosome, clips on, and it reads every 3 base pairs, and that links amino acids together in a certain sequence. That sequence of amino acids then folds and becomes a protein that has a function. Only 2% of your genome codes for proteins. The other 98% doesn't code for those. But what we've increasingly found out is that 98% is incredibly important for when and where that 2% is turned on or turned off. So, like when a human is developing, from like in throughout development, there has to be signals that tells the cells to be skin, to create this body plan. And so that is a really sophisticated system. And so the other 98%, or a large component of that other 98%, is regulating when and where that 2% is turned on and off, when those photocopied memos get made, and under what conditions. And so this is kind of a fascinating thing, is that when we look more broadly and we look at genome size, so 3.2 billion base pairs, humans, and we look at complexity, there's very low correlation, almost no correlation, between the amount of protein coding genes and complexity of an organism, or genome size. Like there's plants like conifers, not saying that trees are not complex, but there's conifers that have 10 times the size of the genome of us, or if not more. I think the biggest genome is a plant, a Japanese plant. It has like, I'm trying to do some quick math, like 80 times the size of a genome of ours.
Wow.
But when you start looking at regulation, that's when more regulation correlates with complexity of an organism. So when and where things are turned on and off. So how much control do we have on the development of an organism? And so that's correlated with complexity, which I think is super cool, because I love epigenetics and I love regulation of genes and things like that. So a gene, most traditionally though, is a protein coding region of the genome. That is generally, and it is made into a very specific type of RNA called messenger RNA. And there's other regions of the genome that get made into functional RNA products, but they don't get made into a protein. Don't know. Yeah.
That's really cool. And I did not know that it was such a low percentage, but it didn't like mean that it was, you know, that the 98% was useless. That's super cool.
Yeah, and
like
it's distinctly not useless, but I remember learning about it before I started university, and there was a lot of people talking about junk DNA. And that's just not the case as we figure out what it does. It definitely has a function. It just doesn't code for a protein. Yeah, nice. Super freaking cool.
Absolutely. So just stemming off of that, I'm curious, how different is my my DNA versus your DNA?
Yeah. So that's, I mean, it's only about 0.1% of the genome that is variable between individuals, which is
Wow.
crazy when you think about like the variation between it. But it's important to remember that 0.1% when you're talking about 3 billion bases
Yeah.
is a substantial amount of variation. And so what do I mean when I'm talking about variation? So I'll introduce the kind of classic, which is mutations or base pair differences. So what that would mean is that in a region of the genome that is the same region between you and me, if we scanned across it, let's say there was a base pair where you have an A and I have a G. That would be what we consider a single nucleotide polymorphism, colloquially known as SNPs. So that is a variant between you and me. So like an example of that would be, for example, I can't actually see what your eye color is.
Wait.
You have blue eyes? You have blue eyes?
They go between blue and green.
Okay, blue and green, but you have light colored eyes. So both
Yes.
you and I have light colored eyes. And so when we think about, or when we talk about eye color, broadly what we can sort them into is dark eyes, light eyes. And then there's obviously the variance between like green, blue, hazel, and things that control kind of color within those things. But if we just think broadly, light and dark, there are genetic mutations in a specific gene that regulates the amount of melanin that you have in your eye. And so people who have light colored eyes broadly have certain genetic variants in that gene, and people with dark eyes have different variants in that gene. Because biology is complicated and we love this like very simple story about eye color, I was doing some reading before this podcast because I just wanted to make sure, get out there. There's a bunch of other variants in different genes. There's also an environmental factor. There's a bunch of things that do change eye color that makes it not just a single gene that codes whether or not you have light color or brown or dark colored eyes. But this is one of the largest determinants about whether or not, one of the genes that contributes the most to determining whether or not you have dark or light colored eyes. So like a lot, we talk about sometimes how much of the variation in this trait can be explained by a single gene or a couple of genes, and a large amount of the variation between people can be explained by mutations in this specific gene, but not all of it, because biology is complicated and messy and
of course
all sorts of things. It's the things that keep me so excited about what I do, but also keep me up at night. So,
fair.
Yeah. And
Fair
so
point.
when we think about protein coding genes, variance in protein coding genes means that the memo has like a different word in the instructions. So when that instruction gets built, the structure of the protein is going to be slightly different because there was a slightly different set of instructions. And a protein that has a bit of a different structure is going to have a slightly different ability to function, or when and where it functions, or things like that. And that's what can create variation in what we see for phenotypes. So when I say a mutation in a gene, that could be one way. It also could be in the regulation of that protein. So let's say it's in the when and where that gene gets turned on. So when it gets copied, there can be mutations in that regulation, and that can also generate variation that we see. So it's not always just mutations in that affect the protein structure. It can also be mutations that change when and where that protein is made.
Oh.
Yeah. So that is what we would consider like regulatory changes.
Okay.
But that, yeah, and those can both be at the genetic sequence level.
Okay.
Yeah.
So that's the genetic sequence level that brings forth variants between organisms, or I guess variants between
individuals of
cells.
the same, like
Yeah.
the same organism, same species.
Yeah.
But it also can be like sometimes when we're looking at evolutionary history, we can look at genetic variants between species that explains why they diverged, how they diverged. Yeah, so
That's
there's
pretty
like
cool.
so there's like the idea that like genetic variants cropped up, it resulted in slightly different phenotypes. Maybe that allowed them to occupy slightly different ecological niches. And then they started diverging because they became very adapted to that specific niche. And we can see that in their genome. We can see genetic variation between these, let's say, like recently diverged species, and go, oh, like that's what controls these phenotypic differences that we see in these species.
That's really interesting. And I think that's a great way to conclude the first segment, because when we come back, we're going to talk about gene expression with respect to the environment. So it's a different way that creates, mutation and things like that. So we'll talk about that when we come back. Okay, we're back. This is segment 2, and we're going to talk about gene expression with respect to the environment. But before we get there, we want to add a couple things, because Brenna feels like it's very important, and I totally agree. Talking offset a little bit, I'm a little checked out because I'm not feeling well, but Brenna explains it very well. I promise you, it makes total sense. It's just it's me. So we want to talk about genetic sequence changes. So Brenna, I'm gonna leave you off with that.
Yeah. So when we think of evolution and we think of natural selection, mostly what we're talking about, and we're really thinking about kind of classic Darwinism, is genetic changes. So sequence changes. So we both have light-colored eyes. I'm going to use this example again. But with someone with dark eyes, they have genetic differences in this gene. Let's say they have, let's bring it back to Punnett squares. They have offspring with someone that has light-colored eyes. There is an opportunity for the genetic information from them and the genetic information from their partner to combine. So there's probability if they, let's say, have the brown type of the gene, and then they also have the blue, because maybe they have one copy of each, and their partner has 2 copies of the blue, there's combinations that their offspring will have dark eyes because it's dominant. So if you have 1 copy, you're going to have dark eyes. Or they might inherit both of the little blue. And so that's one gene, one thing. But this is happening across your genome with variation. It's getting shuffled every time you have offspring. So traditionally, we had thought of evolution really just from this genetic change perspective. And every once in a while, mutations occur in an individual. And that has to happen in germline cells. So there's your somatic cells, which make you, and then there's your germline cells. So talking egg and sperm. For a genetic mutation to be introduced to have a function in evolution, it has to occur in your germline because it needs to be able to be passed on to offspring. And so that happens relatively infrequently because your DNA, as we were talking about it, is the machinery that copies your DNA and does all that is really good at copying it and checks it over and over and over again and tries to fix any mistakes that it made. And so only every once in a while does a genetic mutation occur in a germline. And that is what can get exposed to natural selection and evolution. However, there's also something that's really hit the forefront now and really taken the world by storm. It's something called epigenetics. And so these are changes that alter when and where your genes are turned on or turned off that don't impact the genetic sequence. So these are changes that can happen that are above genome.
Got it.
I don't know if I should just keep rambling about this.
No,
Is there
I
any
like that.
questions so
No,
far?
no. That's, I think
Perfect.
everything's being explained quite well. Epigenetics then is influence based on behavior and environmental stresses.
Yeah, so there's kind of, again, this is another one of these terms that there's about over 20 different definitions of what
Ah,
epigenetics means,
there you go.
depending on what field you're in what regard. So the way that I was taught about it is that it's above the genome, so changes that alter the regulation of your DNA that have to be inheritable. They have to be heritable.
Oh,
In kind of recent years, that has gotten altered to anything that is not a genetic change that can change the way that your DNA is regulated. And that can happen within your lifetime. So they're reversible and they can happen across generations as well. And there's been quite a bit of debate about the transgenerational aspect of epigenetics. But there's like some cool studies when I was an undergrad that I just thought was so neat. So they took rats and you can have genetically identical rats. Their DNA sequence is exactly the same. They exposed some of the male rats to a smell, so a scent cue, and then they stressed them out. So to the point that they did this over and over and over again, so that to the point that the male rats would smell the scent cue and kind of be like on edge.
Mm.
They had their control group that they didn't do this treatment to.
Yeah.
Then they made crosses. So they made a second generation. They used the sperm from these stressed, not stressed male mice.
Mm-hmm.
And they crossed them and made a generation of mice or rats with these two types, with
Right.
genetically the same mom. And
Mm-hmm.
what they found is that the second generation, the mice or the rats that had the sperm from the stressed out individual also responded to that environmental cue. They
Oh,
also
okay.
they were like, they would be startled from that environment, that scent cue. And the male mouse had never met their offspring. Like these are all controlled crosses. So it's not as if the male mouse was like, took its offspring aside and was like, you need to watch out for this scent. It's bad.
Right.
It was something in the like regulation of DNA that had been passed on by not a genetic change. It's crazy. And so
Huh.
what they found is that there was actually a change in the regulation of a gene that is related to stress response. So it was a
Huh.
different pattern of regulation, but it was not a genetic change. There was no
Mm.
changes in the actual DNA sequence. There was just changes in how that gene was regulated. And so it's like
Cool.
these little markers, epi above genome. And
Got
so
it.
like my work looks at gene regulation differences that can get cued by early life history.
Mm.
I don't know if I can talk about my PhD work specifically.
Oh, no.
Yeah, so what I did was I took families of stickleback, the littlest fish,
Mhm.
and I raised their eggs at different temperatures. So everything
Oh,
else was kept the same. Everything was kept the same. Just raise them at different temperatures because what we're curious about is how does an environmental stressor, a temperature stress, affect, like, is there no effect into adulthood? Can
hmm.
they be raised at these different temperatures and has no long-term consequences? Maybe. Or maybe does this stress at early life have long-term repercussions into adulthood?
Hmm.
And so what we did is we raised them at these different temperatures that represent their thermal limits. And then we switched them back to all the same temperature once they hatched. So the only thing that was different in the lifetime of these organisms was what temperature their eggs were reared at.
Mm-hmm.
And as an adult, I actually took their RNA. So I extracted tissue and I isolated all of their mRNA from that. So I basically quantified and determined which copies of the memos were active at any point.
Nice.
And what I have found is that early life history actually changes what genes they have active into adulthood. So there's this signature that there's no genetic change because it's within the lifetime of an organism. There's no genetic change, but there are changes in the way that the DNA is being read and turned on and off, which I think is pretty cool.
Yeah.
Yeah. And so it's just important to think about the differences between genetic changes that can occur, if bringing it back to like genetic changes that occur in somatic cells versus germline cells. Because if you get cancer, unless it's in specific areas, that's not going to affect future generations. Probably.
Got it. Oh, that's interesting.
Because there's genetic changes occurring, but it's isolated, unless it's in reproductive areas.
What kind of cues from the environment? Like for example, what about like UV light exposure, or maybe even high energy or high energy radiation, like say X-ray or a gamma ray burst, or something random like that, or it doesn't even have to. I guess we usually think of like high energy radiation whenever you think of like stressors at the certain
so I
level.
think like that is also because that does introduce potential opportunities for mutations, right? So that it would be actual genetic changes that can occur with mutations. And that's when we think about cancer, like exposure to things that can cause genetic changes. That can have obviously even if we're not talking about evolution, that has very distinct health consequences for an individual, because a certain region or a certain tissue type might get mutated, and that results in, I mean, if I'm just speaking with cancer, unchecked cell growth. And so it's just, there's a lot of regulatory components that monitor when a cell gets duplicated and when it makes another copy and how it behaves, that keeps everybody in check. And when mutations occur in that regulatory system, we can get this unchecked replication of cells. And that can result in things like cancer. And so mutations from like the examples that you used here do have consequences for an individual. Because those genetic mutations can break basically the regulatory or the machinery that checks that everything's happening correctly. So it can introduce mutations that make it no longer function or not function the way it's supposed to. And that has really big problems. And when I talk about though, like environmental cues, it doesn't have to be anything nearly as drastic as that when we talk about kind of just being alive. So for example, like this morning, I woke up, great first step to the day, drank some coffee, I'm sitting on this podcast, I'm gonna go for a run later. These need different states of my being. And those different states need different machinery. They need different proteins, they need different things. Like going back to our house analogy. Let's say going for a run is an environmental cue that you're going to have guests over. And you're looking around your house and you're like, we do not have enough chairs for all these people that we're going to have over. And so then you get a cue saying we're going to have guests over. And so then you make a whole bunch of the photocopied memos on the instructions on how to build chairs. And you make a whole bunch of chairs for when the guests come over in just like your living room, which will be like your muscles, let's say. So then you go for your run, you come back, and in our analogy here, all the guests leave. And you're like, oh man, I don't need all these chairs anymore. But what now I need to do is I need to make something else. So you dismantle all of those chairs, and then it becomes the wood that could be made into something else. And so the environmental cue doesn't have to be anything super drastic. It can just be being alive, because you need different states of being to be able to track even the change that occurs within your day. So there can be changes that occur over your lifetime. As you can think, like a baby needs different information, different processes turned on and off than like as you're going into puberty. Very drastic changes. So the environmental cues and the internal cues as well, it's not just environment, internal cues, say like, hey, we're going to go through some pretty drastic changes. We're going to need to turn on this, turn off that, do this. And then you go into adulthood and this aspect, and then you grow older, and there's different internal and environmental cues as that happens. So that's like over the lifetime, but there's also within your day. Or like if you eat a big meal, your body needs to be able to respond to that. Right. And so what's happening constantly is that your cells and your different cell types are getting information and making different proteins that are necessary to be able to handle what is changing here.
Wow.
Yeah.
Okay.
And that's what I'm really interested in, is how do organisms, species react to their environment at a molecular level?
It's amazing. I guess before we close out on this segment, I kind of want to pick your brain a little bit. We kind of already laid the groundwork for this, and this is probably a dumb question now. With how drastic the climate has changed within the last 100 and so many years, what are you seeing in your field in terms of day to day, week to week, year to year changes?
Yeah, so this is what's like so interesting about it, is that why we're interested in the work that we're doing right now is that how rapid climate change is occurring is that it's exposing organisms to environments that are outside of their historic range, because evolution is really good at making organisms match their environment if it is given enough time.
Of course.
Because genomic, or as we were talking about, mutations happen quite infrequently. And the majority of mutations that occur are going to mess up systems. They're going to, something that I always say is that there's 1,000 ways to break a race car. There's very few ways to make it better. And so most of the changes that occur genetically are going to kind of mess things up. So you're waiting for the one thing that's going to make it better. That being said, there's also just natural variation that happens in the environment. Like as you're saying, we have 0.1% of variation between humans.
Yeah.
Sometimes when the environment changes really rapidly, something that was evolutionarily neutral, so variation between us, let's say we find out that the environment changes rapidly and light eyes are incredibly better than dark eyes. Oh, that's already a variation that we have in the population. So maybe this starts getting selected for the light eye phenotype.
Yeah.
And so that can be kind of the two ways. What we're seeing now with climate change and what we're getting interested in is ways that species and organisms can respond to their environment within their lifetime or within a couple of generations. So this might not be genetic changes. This might be, and this doesn't have to even be molecular, it might be behavior changes. We're seeing in certain areas, species are switching to more nocturnal. They're switching because humans are less present on the landscape at night. And so they're changing their behavior in response to this. And so we can see what we're getting, kind of what we're interested in is understanding at how species might be able to rapidly respond to the environmental change, because it's not happening on the time scale that we would expect traditional evolution or response. Basically, like it's challenging their ability to track the changes because it's happening so quickly.
Right. It's a drastic event relative to what, and I don't even I caution myself on how I want to state this, because there's always the people on the other side of the fence that are looking for a single word to point fingers. But like in terms of natural cyclic approach to climate, this is a drastic event, and so it's challenging a wide variety of organisms.
And it's the time. It's how much change has happened over a very short amount of time. Like, we consider recent evolution 10,000 years. That's very recent evolution. Like, the little species of fish that I study, there's a freshwater and there's a marine type. They diverged about 10,000 years ago. The example of this divergence is considered very recent evolution. So we think about evolution across 10,000 years is considered very recent. And then we look at the changes that have happened in 200 years. The ability of species and ecosystems and to buffer that change, they might have been able to do that if this had happened over 10, 20, 30,000 years, but it's not. It's happening over a blink of an eye. And so this is going to be challenging these ecosystems. And then they also are being challenged by loss of biodiversity, overfishing, fragmentation, logging, industry, all these things. So that they're really, it's not just climate change. It's a whole bunch of other host of things that are challenging an ecosystem to be able to function and species to be able to adapt.
Right.
And I think if I can use this little platform just for a moment, when we think about how ecosystems are changing, we should be concerned from not just like a feel-good kind of way. Our economies, our function, our way of life rely on functioning ecosystems.
Yep.
We get a lot of services, wood. In BC, we had the wildfires, and then we had massive mudslides, took out entire roadways. These are services that we rely on from ecosystems.
Yep.
And it's trillions of dollars if we're just thinking about it from that perspective. So we have a vested interest in maintaining biodiversity and maintaining ecosystem function. So that'll be my little plug, my little side.
I totally agree with that. And I think to have that simmer a little bit, we're going to jump into this next break. But when we come back, we're going to talk about CRISPR, gene editing.
Hard switch.
I know, right? So let that simmer, and we'll be back. Okay, well, this is segment 3. I'm sure you're hanging in here because I think everything's been quite elegantly communicated. We've had some really cool fun facts, some great analogies, and it's really cool information. So I'm sure the people that are watching or listening to this are enjoying their science communication hunger, I guess. Or this part, in my opinion, is my favorite part because we don't typically talk about, like, we'll get to it, but we don't typically talk ethics on this podcast, and it's just something that doesn't typically come up because I don't know, just hasn't. And I like talking about ethics, and we'll get to that. But essentially, we're talking about CRISPR and gene editing. So before, I'm sorry, I might have confused people on why I just said ethics, but we'll get to that. What I do want to start with, though, is, Brenna, what is CRISPR? First of all, it's an acronym, but
it is an
explain
acronym.
That.
Yes. So broadly, which I think is just pretty cool, it's a bacterial immune system. So it was discovered in bacteria because they have this ability to cut DNA when they get infected with something and then store that in their DNA. So it's an DNA editing system.
Right.
That's found naturally.
Yeah, isn't it even more interesting than that? Because it's the tail between bacteria and viruses, and it's been something that's just been happening for a very, very, very long time.
And so scientists saw this thing where it's like, okay, so these bacteria have these systems, and it's called CRISPR Cas9. And so it's these two kind of things that come together that allow for the editing of DNA. And so scientists realized the amount of power and how amount of scientific advancements that could occur if we could use this CRISPR Cas9 system to edit DNA.
Mm-hmm.
And that can sound scary, and we'll get into a little bit more about that. But at its kind of basic is that it can really help us study genetic variants, because we can now experimentally edit DNA. So let's say, for example, let's go back to our idea here. Like, we have two species that diverged recently, and they have this different phenotype. And we think that it is a genetic change in this one gene, this one protein coding. We think it's this that explains the variation in the phenotype. What we can do is we can take this organism, let's say the one that has phenotype A, and we have phenotype B. We could take an individual that has phenotype A, and we could actually use CRISPR to edit the gene in this individual to have the genetic variant that we see in phenotype B. So we can basically test whether this genetic change drives the phenotype for B. Because what we can do is we can take the genetic background of phenotype A, and we can make one change using CRISPR. And then we can see if that now results in phenotype B. And if it does, we have pretty good evidence that that genetic change is what actually drives this phenotype change. And so it allows us to like confirm our hypotheses or provide evidence of our hypotheses of what is the genetics of these different phenotypes, for example, or what is local adaptation, or what are these things? Because bringing it back to our first segment, DNA is very big. And even though there's only 0.1% of variation, that's a lot of different variants. And so there can be a lot of different variants between these two phenotypes, and we can use different techniques to figure out what we might think is the variant driving this phenotype difference. But being able to confirm that or test things with genetic changes is really powerful. So that's one of the ways that we can use CRISPR Cas9 system, is to do very small changes or targeted, they don't have to be small, targeted genetic changes.
Got it.
However, that creates a whole host of ideas about ethics and enhancement and various things, because hypothetically, we could use this to alter individuals if we wanted to. We could use it to change people's DNA sequence.
Right. But before we, I guess, get into the ethical portion of this, there is the aspect that there are wonderful applications to this without even approaching an ethical conversation. What kind of applications might that be, just for someone who's not familiar with CRISPR?
Yeah, so I mean, and CRISPR is the system in and itself, and it's fascinating in and itself. But really what we can do with it is of interest with people. So we can do genome editing. So an example that I see here is, like, for example, something like sickle cell anemia. So sickle cell anemia is a mutation in the genome that results in your blood cells having this sickle shape.
Right.
And so that sickle shape doesn't transport oxygen, can make people very, very sick.
Yes.
And so that is a mutation that happens in a protein coding region of the genome that breaks something. It's a loss of function. I'm pretty sure that it's a mutation that occurs. Using CRISPR, you can introduce a functional protein. So you can basically edit the genome so that it no longer has the protein copy that has the broken version. You can basically find and replace, Command F and replace that region of the genome with a functional product. That being said, as we were talking about earlier, you have trillions of cells. So once you are a full-fledged adult, or you are kind of out, you have developed, you're out in the world, you have lots and lots of cells. So the challenge becomes, how do we edit the DNA in all of those cells? how do we try to get that information into all of your cells? It's a lot. And then we're still working on that kind of challenge. But the thing is that, for example, something with sickle cell anemia, you don't have to actually target all of the cells. It turns out there's actually kind of a threshold amount that you can target. And I believe that they're targeting cells before, I was talking earlier about differentiated cells and non-differentiated cells. They're targeting a certain section of your cells so that it can copy, and when they copy, that information gets put into all of their like daughter cells.
Got it.
And there's kind of a threshold where not every single one of your cells has to be edited for you to be treated for sickle cell anemia. There just has to be a certain amount that they're able to do. And so in that space, CRISPR has the ability to be incredibly powerful to treat disease.
Yeah.
And that is treatment of somatic cells. So that is not germline. It's not going to be passed on to the next generation. It is just treatment of an individual. And it is a way of potentially treating a genetic disease at the source rather than treating the outcome of the disease. So without treating kind of like, without managing the symptoms, it would be actually treating the genetic disease at the actual source. And so it's incredibly powerful in that space and has the ability to change people's lives in that area if it's within generation of a genetic disease.
Right. And then what needs to be carefully examined moving forward is then the germ cell aspect of
and
the germline aspect.
Also genetic modification of individuals. There's been the kind of line is, are we treating a genetic disease or is it enhancement?
Yes.
So
agreed.
There's a pretty firm line agreement that CRISPR should not, and gene editing as a whole should not be used for enhancement.
Right.
For treatment of disease, that the ethics behind that is that we should be, or we should. That being said, really generally it's into germline, because then that would be changing someone's genetics that gets now passed on to the next generation. So it wouldn't be just treating of an individual of their disease state. This would be like influencing evolution and influencing reproduction. And that starts to get into some pretty not so great conversations about ethics and right and wrong and what we should be messing with and what we shouldn't be messing with.
Right. Because like, as you said earlier, like the expression of certain characteristics can involve many genes. And whenever you're attacking germline, it could end up that maybe we don't fully understand something and then something bad happens, and now it could potentially be carried because now you don't want to tell them, oh, well, you can't have offspring because we've tickled with your germline, like you don't want that's unethical.
And that's not even it's we just wouldn't know potentially the downstream ramifications. And it also is just will open the gates of a potentially very slippery slope of what becomes treatment of disease and what becomes enhancement in eugenics and various things. And so it's really that as soon as we open that door of we are able to alter genetic sequence that can be inherited into next generations, that's just it's a slippery slope of what we would consider okay and not okay. And so it's kind of just the general consensus is that maybe it's just not a slope that we should get on at all. Like maybe this is just not this, that is just not going to be the hill we want to be on. Like
definitely not.
Using it to treat a genetic disease within the lifetime of an organism, in the lifetime of an individual, that has incredible power and ability to change people's lives and be used as a therapeutic treatment.
Yeah.
When we start getting into the idea of genetic changes for reproduction or for offspring that could affect offspring in that space, that starts to open some kind of like doors and thoughts that is just deeply not ethical.
Oh yeah. There's those issues of eugenics. There's the issues of socioeconomics. There's the issues of geopolitics. Because
there's access to. Yeah,
go
sorry.
ahead.
I got excited, so I interrupted you. My apologies. There's also conversations about the access to these kind of treatments, the access to this, and that is actually a conversation about ethics, about even CRISPR used as a therapeutic tool, because it's still quite expensive, and how are we going to allow that this is available to everybody? And so that's I can't say that I'm an expert in that in any way, shape, or form, so I won't really weigh into it. But from a gene editing standpoint, I think that if we start playing God, it becomes difficult.
Yeah, the playing God is
the power of an individual.
And
experiments like this have been shut down already.
It was after the 2020 Nobel Prize award, I think, but it was a group of Chinese researchers that were messing around with germline, and they were trying to treat a disease, like a legitimate disease that everybody would go, oh yeah, that's a disease to treat, but it was shut down by no journals will publish it, and the science community just kind of was like, no.
Yeah, because the thing is that it is an interesting conversation about ethics, because it's a disease state. We could cure this, we could do it, but it just really opens up the ability, if we start altering in germlines, for conversations about designer babies, and it's just not a space that currently or probably ever we really would want to explore. But from a CRISPR, from a research perspective of being able to test hypotheses about genetic mutations and what they mean and how that looks for evolution, and also for therapeutics of treatment within the lifetime of someone, it is. There's a reason why it won the Nobel Prize quite recently, very recently, which was so exciting for us. So from that aspect, CRISPR is so freaking cool. But we do have to be careful about the potential for it to be misused.
Right. Do you think that there could ever be a point, and this is obviously extremely hypothetical, and I'm against the whole,
feel like you're
I'm
setting
playing
me up
devil's
for failure here.
No. Devil's advocate. This is what is it? The doppelganger of Sam. Do you think there could ever, and there's nothing wrong with this, but do you think there's, in terms of bringing it up, but do you think there could ever be a point in which we do have a geopolitical standpoint where we say there are these 20 diseases that we would like to eradicate, and we have the technology now? We don't have the technology now based on what I've read or what I've heard. But if we had the technology and we had the geopolitical agreement on a set of diseases, then would it be acceptable to move forward? Yeah.
I think it's interesting because when we think of different types of diseases, so for example, if we were very clear about what they are caused by, so let's say it's a mutation that is a loss of function mutation, so it breaks the protein in this area, and that causes disease state. I still don't believe that maybe this will be thousands of years in the future when we've done more and more research, because biology is incredibly complex and it's messy, and it interacts with the environment, and It's just, I don't think in our current understanding that this is something that we want to open. I don't
No.
think it's something that we have the knowledge about to confidently say, we are only going to eradicate these diseases and there will be no other consequences to this.
Agreed.
Because it's just our understanding of how genes work. And I don't want to like make it sound like we don't have any idea of what's going on. Just it's so complex, and we're chipping away and understanding, and we understand a lot about how genes work and their interactive effect and into environment. But we just don't, there's still so much we don't know. And so I think that if we started opening up that, looking at that space and making that a possibility, I just don't think currently we have the ability to confidently say that by altering these things, we would just eradicate this disease without there being any other consequences.
Yeah.
And I also just think that that is morally or ethically, I don't know. It's an interesting one. It's an interesting, because obviously don't want people to suffer and want people to be genetically healthy. But I do think that if we could focus more on the treatment of individuals within their lifetime, so that in individual, when this crops up and when they can be treated for it in that aspect, that eliminates some of these ethical and moral concerns and still allows for individuals to be treated and to have a high quality of life and not to suffer in ways that they don't have to. So I think that the focus probably being on within generation treatment and therapeutics is just probably a better focus.
I agree. And I think my response would be that there, just like how, obviously this is a very, very far parallel example, but like we're maybe in the US, we're developing, there's companies that are interested in developing ways of artificial intelligence, like ethics. And so for me, it should be a private effort, but it should be a political and a geopolitical effort to instill committees or companies that delegate the regulation of our ethical concerns that we're bringing up here.
Yeah, there has to be legislation. There needs to be governing bodies that are able to keep things in check.
Yeah, because I think it's way more important. Obviously, it's really important to cure people, but like to work in tandem in that manner is just so important.
Yeah. And I feel like we're but it's a fascinating conversation. It's and it's an important one to consider, and CRISPR, and I think it's also important to talk about, maybe if you don't mind, about some of the limitations about CRISPR, because I've seen a lot of misinformation and people posting photos that are of like a chicken with wings with dog legs and being like CRISPR, not biologically possible, not possible. You're like, even if you put the dog leg components of the genome, which don't exist because it's, again, this complex regulation of on and off, that organism wouldn't, because you would have to do this at the embryo stage. That you would have to do. Talking about this very hypothetical situation that cannot exist, you would have to do that. Like, you would have to take the DNA of a chicken and the DNA of a dog, and you'd have to put it together and then make this, or it just wouldn't survive. It wouldn't even replicate to make multi-cells. It would just not make it, because these are very divergent genomes. Like the machinery wouldn't understand the instructions. Nothing would make sense, and it would go, I'm going home, I quit. And it just wouldn't work. And so when you see things like that, of like being like, we've melded these things together and all this, no, they haven't. That's not how that's not how CRISPR works. And it's not how gene editing can work. Like there has to be a lot of like compatibility, making sure that when you're doing like, you're moving a gene from two very closely related species or like ecotypes of the same species, that moving this one gene and putting it in that doesn't disrupt things that are vital for survival. And that's between species that are very similar, and you're talking about a single gene. If you just mashed a dog and a chicken genome together, it just wouldn't work. It biologically can't exist. And so I just have seen that a few times actually on TikTok. I've been tagged in videos where people are like, can this happen? I'm like, no,
not even close.
Not even close.
A good way for, I think a good layman perspective is that the genome of a Homo sapien and the genome of like an orangutan is like, what, 1% difference? Maybe I'm probably even maybe under exaggerating that by a little bit, but we obviously can't breed with them.
Yeah.
And that's 1%, but like between you and I, it's like 0.1%.
Yeah.
Right? Imagine a dog and a cat. Like, did you ever see a dog come from a cat? No, because it's
So imagine if you
extremely different.
Just, like, artificially mash those genomes together using CRISPR. One, it just doesn't work like that, but in general. But there's just no way for that to happen, and like that. So if you're ever tagged, or if you ever see a video come across your For You page and it's got a dog with chicken wings on it, it's just Photoshop. Or someone has taken their poor pooch and glued some chicken wings on it. And the dog's going, I don't think I want to be part of this. So it's not genetics. It's nothing like that. We're not there yet. And we're never going to be there because it just doesn't work.
Just don't work like that. You just can't do it
like that. No.
So I think I want to open the floor to you, Brenna. Do you have anything else that you want to talk about before we get on out of here?
You know, I can't think of it. It's been a wonderful conversation to be talking to you about this, about something that I love. We really covered a whole bunch to be able to go from, what is DNA to talking about CRISPR. Is there anything that, I'm going to flip it around on you. Is there anything, do you think, that I was unclear about that I could help better explain or go over again or anything like that?
Not from my point of view. I'm sure that. So now I'm gonna do this. I'm gonna point into a different direction. I guess we're playing ping pong, but instead it's a third-party ping pong serve. So if anybody does have any questions for you from this podcast, how could they reach out to you?
Oh gosh. Well, I mean, I do have a TikTok account. I believe it's Biology Bren. I'm not particularly active on there right now. I am hoping to come back to it. Turns out finishing your PhD is a lot, and I just haven't had a lot. But if there's specific questions that anybody has, sending me a message on there, and I would happily make a video about it or answer. I have a website. You can contact me through that as well. Read my papers. No, just plugging my papers, please. Yeah, I mean, so really I would say those kind of two major ways with my website and my TikTok. I do talk about, there's some videos still on there about evolution and for example, like I made a video a while back about whether we actually share 60% of our DNA with a banana. Talk about what that actually means.
That's good.
Spoiler, we kind of do. We don't really. It's
it's complicated.
Turns out it's a specific part of the DNA. Yeah, so I'd say that those are probably like two ways the best, and I'll try to answer any questions. I'm not always the most responsive on TikTok, but I am trying to get better because I do love that part of communicating science. Yeah.
Well, I think that is a good way to end the podcast. That way that they can be like, oh, fresh in my head. Go talk to her. Okay. Yeah. So, Brenna, Thanks a bunch for being on the podcast. This was
Thank you for
wonderful.
having me. This has been absolutely delightful.
And that concludes this episode of Everything STEAM. I just wanted to take a quick second and thank Brenna for sharing her knowledge and expertise about, you guessed it, genetics and, of course, CRISPR. To follow Brenna and her work, I suggest that you visit her website, brennastanford.weebly.com. I would also love to mention my amazing team for their collective efforts to make this show happen. This podcast was edited by Ariel Piermont, QC by Pandypit Eriksit, and the episode art was created by Gabrielle Edmiston. After the episode, please give our podcast a rating and review on whatever platform you get your podcasts on. We're always looking for feedback, and the rating would greatly help us out in the fight against the algorithms. Lastly, be sure to check us out on all the socials for podcast news, upcoming episodes, and fun STEAM content. Just search Everything STEAM on Instagram, TikTok, and Facebook to join in on the fun. Once again, thank you for listening to Everything STEAM. I'm your host, Sam Stanford, and as always, stay curious.